A brain is made up of the tissues weighing only about 1.3 kgs but consists of infinite neural connections which control all our behaviors. The human brain is highly complicated. It is an organ that is responsible for so many varied tasks and activities ranging from simple to complex, and still it is the least understood organ of our body. The scientific study of the anatomy, biochemical processes, and the physiology of the nervous system is known as neuroscience. The scientific study of anatomy, biochemical processes, and the physiology of the nervous system is known as neuroscience. A closely related field of the neuroscience is biopsychology, which draws information from it to study human and animal behavior. It is also known as the psychobiology, behavioral biology, or behavioral neuroscience. In this unit, we will discuss about this related branch of neuroscience, that is biopsychology. Major divisions of the biopsychology and the various methods employed to study it. Biological approach to the study of the human and the animal behavior is known as the biopsychology. The biopsychologists try to investigate scientifically how biological processes interact with cognition, emotions, and other psychological processes. We can trace a long history of the study of biology of behavior, but biopsychology as a separate discipline of neuroscience emerged in the 20th century. D.O. Habs, Canadian psychologist, seminal publication. The Organization of Behavior 1949 in the field of the psychology and the neuroscience paved the way for future investigation of neural foundations of the behavior. What Hebb proposed in 1949 is being applied in the present times in the fields of engineering, robotics, psychology, neuroscience, and neurophysiology. Biopsychology thus draws information from neurosciences and uses the information to study human and the animal behavior. It may be better understood as that the neuroscience is a team effort and biopsychologist is a part of team. A biopsychologist may draw the study of behavior. Few of the disciplines of the neuroscience that are of particular relevance for biopsychology as are follows. Neuroanatomy, it is the study of the structure of the nervous system. Neurochemistry, it is the study of the chemical aspects of nervous activity. Neuropathology is the study of the disorders of the nervous system. Neuroendocrinology is the study of the interaction between nervous system and endocrine system. Neuropharmacology studies the impact of drugs on the neural activity. Neurophysiology studies the functions and the activities of drugs on the neural activity. So, it may be rightly said that the field of the biopsychology is diverse and it is one of the branches that contributes to neuroscience. The research conducted in the biopsychology is also from varied perspectives. Biopsychological research is conducted on the human or non human participants. The most common non human research participants are mice, rats, followed by the cats, dogs, and primates. There are three advantages in conducting research on non human participants. The first being that the brain and the behavior of non human participants is less complex as compared to human participants, thus, the basic fundamental brain behavior interactions may be revealed. Secondly, conducting research on such a group leads to a comparative approach where biological processes of different species may be compared. Thirdly, it is easy to conduct research on laboratory animals rather than human participants due to ethical reasons. That is, there are fewer ethical constraints while we conduct research on animals. Though the research in psychology and its branches are all governed by the strict ethical guidelines formed by the organizations like American Psychological Association, guidelines for the ethical conduct in the care and the use of the non-human animals in research, and the Indian Council for Medical Research, guidelines for biomedical research in India. Research in biopsychology may be a pure research or applied research, and the studies based on the empirical investigation with applications of the society. The research can be done using an experimental approach or non experimental approaches or case study. The scope is to understand the basis of behavior. Behavior changes are primarily due to disruptions in the various brain regions and neural circuits. This may happen because of the head injury. Secondly, learning, exercise, etc. also changes the neural structure. So, how it produces a corresponding change in the behavioral functions can be studied with the help of various research methods that are used to understand the functioning of the mental processes by examining the biological systems. Biopsychology also aims to understand aspects like the evolution of the brain and and its influence on behavior, the development of the nervous system across the lifespan, which areas of the brain are involved in the sensation, perception, memory, movement, the role of the brain in emotional expression and regulation, language, and the brain damage or trauma. It also seeks to understand the role of genetics and endocrine system in maintenance of homeostasis and enhancing the health and well-being of the people suffering from various neurological disorders. There has been some very significant research related to the brain behavior interactions that have been awarded Nobel Prize. You must have read about the Ivan Pavlov, a Russian physiologist. Pavlov was Nobel Prize winner in the 1904 for his research on physiology of digestion. Pure research, it is also known as the basic or the fundamental research. It is conducted without any prior aim or goal. It is exploratory in nature and driven by the intuition and interest. Applied research is conducted with the prior aim. Specifically, it starts with the aim to solve an existing problem. Thus, it is descriptive in nature and has practical implication. This type of research is mainly conducted in technology, medicine, and agriculture. Then we have the divisions in the biopsychology. You must have learned about the nature and the scope as well as how research is being conducted in different ways in the area of the biopsychology. There are few approaches that gain prominence and consequently separate divisions of the biopsychology emerged. Biopsychology has six main divisions such as physiological psychology, psychopharmacology, neuropsychology, psychophysiology, physiological psychopharmacology, neuro, psychophysio, and cognitive neuroscience, and comparative. These are the divisions of biopsychology. So these approaches are overlapping and many biopsychologists follow more than one approach. Physiological psychology uses controlled experimental conditions to stimulate the brain and study its effect on the behavior. It involves different manipulation and recording of the brain by primarily using surgical or electrical stimulation on animals for research. Their aim is to develop theories about how neural mechanisms control behavior. Psychopharmacology uses drugs to stimulate neural systems and then observe their effects on behavior. The purpose is to study the interaction between brain and behavior, but majorly the experiments are applied in nature, focusing on developing therapeutic drugs and reduce drug use. Pharmacologists uses drugs to stimulate neural system and then observe their effects on the behavior. The purpose is to study the interaction between brain and behavior, but majorly the experiments are applied in the nature, focusing on developing therapeutic drugs and reduce drug use. Neuropsychologists study the patients who have had some head injury or trauma and are suffering from brain damage. The neuropsychological test helps to diagnose the impairments and work out a suitable treatment for dealing with it. This subdiscipline especially uses case studies and the quasi experimental studies of the patients with brain damage caused due to disease, accident, or neurosurgery. The focus of these studies is cerebral cortex, the most prominent part of the mammalian brain, which consists of cellular layers on the outer surface of cerebral hemispheres. It's a patient who have had some head injury or trauma and then suffering from the brain damage. It is to help diagnose the impairments and work out a suitable treatment for dealing with these. This subdiscipline especially uses case studies and the quasi experimental studies of the patient with brain damage caused due to diseases. It consists of the cellular layers on the outer surface of cerebral hemispheres. Then we have the psychophysiology. So psychophysiologists use non-invasive techniques. The physiological activities recorded from the surface of the body to examine the relationship between the physiological activity and the psychological processes as attention, learning, memory, emotions in human participants. Various measures that are used are like the scalp electroencephalogram, E E electroencephalogram, electroencephalogram, muscle tension, eye movement, galvanic skin response, GSR, heart rate, blood pressure, and pupil dilation. Then we have the cognitive neuroscience. Cognitive neuroscientists study the neural basis of the cognition and higher order cognitive processes like thinking, memory, attention, as well as the perceptual processes. Hence, human participants are taken for research. The techniques used are non invasive. Functional brain imaging is the main method of the recording the brain activity. The comparative psychologists study the behavior of different species to understand their behavior from evolutionary, genetic, and adaptation perspective. Behavior is studied in the either laboratory condition or it may be observed in its natural environment. The latter is known as the ethological research. Etho. Behavior is studied in either laboratory condition or it may be observed. Okay. It is known as the ethological research. Then the methods to study biopsychology. The preceding section introduced you to the nature, scope, and the major division of the biopsychology. This section deals with the methods employed to study human brain. The structure of the brain is quite complex and understanding its working is all the more challenging for the researchers. With the discovery of the Broca's area, it was concluded that the different brain areas had different functions. Broca's area. Broca's area. It is concluded that different brain areas had different functions. In 1861, Paul Broca, French neurologist, found that one of his patients who had lost the ability to speak had damage in the left frontal cortex. Left frontal cortex. More patients with the loss of speech show damage in and around this area, which is known as Broca's area. Broca's area is the left frontal cortex, and patients with the speech impairability shows damage in this area. Thus, the research conducted in these areas conclude that the brain damage causes effects like increase or decrease in hunger, changes in the emotional responses, to mention just a few. We have the methods to study the study to use which are used to study of the brain, which are first is the ablation method. Ablation means removal of a brain area, generally with a surgical knife. It is different from lesion. Both ablation and the lesion methods are invasive and involve cutting a part of brain. Ablation method thus involves the destruction of some tissue in the brain. This is followed by assessing the changes that occur in the behavior due to the removal or the destruction of the tissue. Any wound or injury thereby is known as a lesion, but one has to be very careful as different parts of the brain are well connected to one another and work in conjunction with the another. So if a lesion is created in one area, it may exert an influence on the neighboring area as well. Sometimes animals also recovers from the brain damage, either partially or the functions of the damaged part of the brain are covered up by the another part of the brain. There are different ways to cause lesions. The main techniques discussed here are aspiration, radiofrequency, knife cuts, cryogenic blockades, and the nerve poison. Okay, so ablation method does involve the destruction of some tissues in the brain. This is followed by assessing the changes that occur in behavior due to removal or destruction of the tissue. It is different from lesion. Ablation means removal of a brain area, generally with a surgical knife. Okay, both ablation and lesion methods are invasive and involve cutting a part of brain. Ablation method thus involves the destruction of some tissue in the brain. This is followed by assessing the changes that occur in behavior due to the removal or the destruction of the tissue. Any wound or injury thereby is known as a lesion. Okay, so if a lesion is created in one area, it may exert an influence on the neighboring area as well. Sometimes the animals also recover from the brain damage either partially or the function of the damaged part of the brain are covered up by the another part of the brain. There are different ways to cause lesions. The main techniques discussed here are aspiration, radiofrequency, knife cuts, cryogenic blockades, and nerve poison. Aspiration. These are the methods which cause lesions. Lesions are the basically any wound or injury caused during the removal of brain area, which is the ablation. So methods which cause lesions are aspiration, radiofrequency, knife cuts, cryogenic blockades, nerve poison. Aspiration, a fine thin needle is placed on the tissue which can be seen by the surgeon and accessible by the instrument that needs to be examined. And it is aspirated and the contents are then placed on the slide and examined for any diagnostic analysis. This is generally the preferred technique when a part of the cortical tissue has to be sucked to reveal the area that is under it for the further investigation. Basically, a fine thin needle is placed on the tissue and the contents are then placed on the slide and examined for any diagnostic analysis. Generally, the preferred techniques when a part of the cortical tissue has to be sucked to reveal the area that is under it for further investigation. Then we have the radio frequency. RF current is used to produce subcortical lesions. The wire is placed on the tissue which needs to be examined. Radio frequency devices produce a high frequency current that is alternating current. The heat that results from this current destroys the tissue in that area. The duration and intensity of the current will determine the size and the shape of the lesion. So radio frequency current is used to produce subcortical lesions. The wire is placed on the tissue which needs to be examined. Radio frequency devices produces a high frequency current that is alternating current. The heat that results from this current destroys the tissue in that area. The duration of and intensity of the current will determine the size and shape of the lesion. Then we have the knife cuts. Sectioning is used to make a cut on any area of the nerves of the brain. There is a special device that is used to make subcortical knife cuts very carefully by placing the brain using a stereotaxic apparatus. By using such a technique, extensive damage to the surrounding areas decreased. Stereotaxic apparatus is used to make a cut on any area of the nerves of the brain. Then we have the cryogenic blockade. This is a temporary way of inducing lesions. So cryoprobe is placed in the brain. Through this tube, a coolant is sent to the area under study. The coolant reduces the temperatures at its tip to such an extent that the neuron stops sending impulses. Since the temperature is maintained at above freezing point, there is no structural damage. Once the temperature is restored to the normal level, the neurons start functioning from the neuron starts functioning again. Nerve poison, certain chemicals such as ascanic acid or the ibotonic ibo are used as nerve poisons to create lesions in a particular area in the brain. These chemicals are inserted to a glass tube known as the cannula. The destruction is only partial, it is only the cell body that is destroyed. So if there is any change in behavior, then that can be attributed to the cell body. And we have the histological methods. With the advent of technology in the present time, there are many techniques and the methods to study anatomy and the functioning of the human brain. For studying minute details of the tissue, microscopic investigation histological methods are used for the purpose. In this method, the sample of the brain that is to be studied under investigation needs to be prepared so that it can be mounted on a slide and observed under a light microscope. The neural tissue is very fragile and under normal condition it may just disintegrate. Hence the tissue needs to be fixed and stained in order to be viewed on the slide. When it is taken out, the bacteria or molds tend to decompose the neural tissue. Fixation. In this method, a fixative is used to fix the tissue. The tissue is cleaned and dipped in a fixative solution. The most commonly used fixative is formalin. This fixative prevents autolysis and makes the tissue hard, but still the tissue is not so hard to be cut using a microtome. Automatic slicing machine used in the microscopy to produce thin section of the fixed and the embedded tissue for microscopic observation. Hence, the brain tissue is free so that the tissue becomes more hard. It is also used to stabilize neural tissue. Hence, the brain tissue is free so that the tissue becomes more hard. It is also used to stabilize neural tissue, but still, the tissue is not so hard to be cut using a microtome. Automatic slicing machine used in microscopy to produce thin sections of the fixed and embedded tissue for microscopic examination. So, microtome is the machine used in microscopy. Hence, the brain tissue is free so that the tissue becomes more hard. It is also used to stabilize neural tissue. We have the term autolysis. Auto means self. Thus, autolysis is the self digestion or breakdown of the cells in animals, plant or microorganism tissue by their own enzymes. Freezing the tissue needs to be frozen in order to be sliced. To freeze the tissue, it is dipped in a sugar solution and then put in a freezer. The The temperature is regulated in a way so that the tissue is maintained at an appropriate temperature. Once the tissue is hard enough, it is to be placed on the microtome to be sliced. Then we have the embedding. Another way to make the brain tissue hard is by embedding and covering it by paraffin or nitrocellulose. Nitrocellulose. Once the tissue is hardened, it is cut using a microtome. These slices are then placed on a slide and mounted on it using albumin, which is taken from the egg. The slide is dried, it is then dyed using different chemical solution. Then we have the staining. It is done so that the structural details of the tissue becomes apparent under the microscope. It is done by darkening or coloring particular feature of the tissue. Nasal stain or cell body stains are most frequently used to dye the cell bodies. Myelin stains color the myelin sheet to study the nerve pathways. Membrane stains like the Golgi Cox stain helps to color the cell body, dendrites, and axon. We have the psychophysiological recording methods. These are the methods that help to examine human participants in a non invasive way. Such methods record from outside the skull and the other parts of the body without inserting anything. The most widely used methods in this category are electroencephalography. The electrical activity of the brain is recorded using electroencephalograph machine, and the technique is known as the electroencephalography. Electrodes are placed on the scalp of the patient and recording is done on the computer. Earlier, a rolling paper was used where the readings were displayed. Now, a computer printout is taken. The EEG signals help in the diagnosis of the various disorders of the brain: mild head injury, cerebral pathology, epilepsy, and the speed of the processing information, and how the memory functions with an increase in the age. The EEG signal helps in the diagnosis of the various disorders of the brain, like the mild head injury, cerebral pathology, epilepsy. Then we have the magnetoencephalography, also referred as the MAG, measures the changes in the magnetic fields on the surface of the scalp produced by the changes in the basic patterns of the neural activity. The muscle tension, we have the electromyography. The electrodes are attached to the skin over the muscle that is to be investigated. When the muscle contracts, producing muscle tension, it is recorded. This information helps us to know about muscle tone. Electrooculography, the eye movements are recorded by using the electrooculogram. There is a slight difference in the electrical potential in the back and front portion of the eyeball. This difference in potentials is recorded with the help of electrodes that are placed near the eye. The eye movements are recorded by using the EOG. Eye movements. Then we have the electrodermal. The two main indices of the electrodermal activity are the skin conductance level and the skin conductance response. Electrodermal recording. Skin is recorded by attaching the electrodes on the fingers and recording the fluctuations in the skin conductance. SCL measures the background level of skin conductance associated with a particular situation. SCR is the transient change in skin conductance associated with the distinct experiences. When there are different kinds of emotional stimuli, variations are produced in the skin conductance level due to changes in the sweat gland activity. We have the electrical stimulation. The brain can be stimulated electrically to gain information about the working of its various neural systems. When the brain is stimulated, there is a change in the resting membrane potential and an action potential is initiated. This causes behavioral changes depending upon which part of the area has been stimulated. For stimulating the brain electrically, electrodes are used. Microelectrodes are made for the fine metal wires through which the nerves are electrically stimulated. Macro are used to steel wires and are placed either on the top of the scalp or inside the brain to record the functioning of the tracts, which are large number of neurons together. The electrical recordings can be made over a long period of time, such as when the animal is recovering from any surgery. This is known as the chronic recording. If the scientist is focusing to study a particular nerve pathway or where the information is going, then acute recording is done. These recordings of the electrical signals are then displayed through an oscilloscope or ink writing oscilloscopes. Oscilloscopes. It helps to display the electrical recordings per unit time. It has a cathode ray tube on which the display is done. It is used to record the activity of the one single neuron. When the action is detected, it is amplified and easily heard by the researcher. Okay, then we have the ink writing oscilloscope. It is also known as the polygraph. The evoked potentials are recorded with the help of the various electrodes that are attached to the scalp. Polygraph. There are needles, pen tips that record the neural activity on a rolling paper with ink. When the signal is detected, it is amplified and the pens move up and down, writing with the action in the ink or continuously rolling paper. These recordings of the brain are known as the electroencephalogram. EEG are the recordings of the brain. Recordings of brain. This helps to detect any tumor in the brain, epilepsy, or study sleep and arousal pattern. Modern techniques use computer to record the information and display it on the screen. Computer helps to display, store and analyze the information that is collected by recording the electrical signals.